Hello, everybody, and welcome to the Frontiers in Science Deep Dive webinar. I am Laure Sonnier. I am the executive editor of Frontiers in Science, Frontiers' flagship multidisciplinary journal, publishing exceptional transformational science for healthy lives on a healthy planet. So today's deep dive is on harnessing agri-food system microbiome for sustainability and human health. It presents Frontiers in Science lead article led by Professor Paul Cotter and colleagues on hidden connections between our food, our health, and our planet, looking at microbiome-based solutions to help create a more sustainable food system globally. First, I would like to thank warmly Professor Cotter, Dr. Kostic, and Dr. Fernández Gómez, who are all here with us today for bringing this important transformational work to Frontiers in Science. We are absolutely delighted to give it a home. So before we meet these authors and our guest experts, just a few words about Frontiers in Science. So Frontiers in Science, as I was saying, is our flagship multidisciplinary open access journal at Frontiers. The journal focuses on high-impact transformational science to help accelerate solutions for healthy lives on a healthy planet. And the way we do it is that we commission exceptional peer-reviewed lead articles from world-leading researchers. And then around each of these articles, we built a unique hub of multi-audience content, which includes editorials, scientific viewpoints, policy articles, and content for children and the general public. And then we maximize the dissemination of the hub and content via large-scale communication campaigns. So the project in itself, the journal in itself, is all designed to really extend its reach and impact across science, policy, and society. And to date, launched in February 2023, we have now published 23 hubs spanning 87 articles, covering a diverse spectrum of transformational science. And this science has been viewed over 2 million times already, downloaded around 300,000 times, and cited around 800 times, and has been also regularly picked up in top media. So this is really a testimony of the broad impact Frontiers in Science is already having. And we believe that really no other publisher does this actually in the way we do it. And we are really happy to be driving innovation in publishing so that critical transformational open science reaches everyone and can help accelerate scientific progress for all. So today's discussion is going to be moderated by my colleague Rui Fernandes, who is the journal manager at Frontiers in Science, and I'm now delighted to hand over to him now. So thank you all, enjoy the event, and Rui, over to you.
Thank you, Laure, and hello everyone, and thank you very much for joining us today. My name is Rui Fernandes. As Laure was mentioning, today we are focusing on the lead article from Frontiers in Science, Harnessing Agri-Food System Microbiomes for Sustainability and Human Health, which was published in July of this year. Why are we discussing this theme? Over the last few decades, we've becoming increasingly aware of the importance of health-promoting bacteria and other microorganisms, prompting wider availability and consumption of probiotics and fermented foods in the hope of health benefits. These food system microbiomes form a complex, dynamic, and interconnected network that impacts both human and environmental health. In their Frontiers in Science lead article, the authors map our current understanding of the microbiomes in food systems, known as agri-food system microbiomes. They do this using biochemistry techniques called omics and produce a comprehensive snapshot of the whole system. The authors then call on scientists, food producers, educators, regulators, and consumers to protect our microbial connections. You are now seeing the page of the lead article. The lead article is also published together with content from multiple sources in an article hub, and this hub includes a viewpoint article by Dr. Fanette Fontaine and Karel Callens of the United Nations Food and Agriculture Organization, FAO, another viewpoint article by Professor Michael Gansel of the University of Alberta and colleagues, and a policy outlook by Julie Barrett O'Brien and colleagues at the OpenBiome Foundation. You can also find in the hub a lay explainer, infographics, and a video, as well as an article in Frontiers for Young Minds, our journal for kids. Before we hear from our experts, here's our program. Professor Paul Cotter will set the scene for today's discussions, introducing food system microbiomes and recent advances in our understanding of them. Dr. Paula Fernández Gómez will dive deeper into examples of microbiome interconnections and interventions. And finally, Dr. Tanja Kostic will discuss how we move from deeper understanding to application and impact. After these presentations, there will be a panel discussion with two additional guests, Karel Callens of the United Nations Food and Agriculture Organization, and Julie Barrett O'Brien, CEO of the OpenBiome Foundation. All right, so now let's hear from our authors and experts. I'm delighted to welcome Professor Paul Cotter. Paul is an international recognized expert in molecular microbiology with a particular focus on food microbiology, especially fermented products, probiotics and postbiotics, the food supply chain, and the human gut. He is also the head of the food biosciences in Teagasc, the Agriculture and Food Development Authority in Ireland, and serves as a principal investigator and leading research at leading research centers, including APC Microbiome Ireland, Vista Milk, and Food for Health Ireland. Paul is also head of microbiology and co-founder of SecBiome, a microbiome sequence and bioinformatics service provider. The author of over 450 peer-reviewed publications, Paul is also the field chief editor at Frontiers in Microbiology and recipient of numerous awards, including an honorary doctorate from the University of Antwerp. His work is defined by a commitment to advancing food and health through microbiome science. Paul, thank you very much for bringing this article to Frontiers in Science, and the floor is yours.
So over the course of the next few minutes, I'm going to set the scene, as mentioned by Rui, so not get into too much details. So the more detailed stuff will be provided subsequently by Paula and Tanja. But rather, for those of you that maybe are less familiar with microbiomes, our food system microbiomes, to introduce you to them and maybe explain why we know so much more about them in recent times. And then subsequently, my colleagues will tell you more about the interconnectedness of all of those various different environments. I suppose the first thing to appreciate really is that microbes are everywhere in the food system, right across the full system, from soil and marine environments all the way through to the human gut after we consume foods at the other end. And microbial communities are essential for food system health, for the security of the food system, and also for climate change mitigation. So having an in-depth appreciation of what's happening with regards to those microbiomes is really of great importance. So generally, among the public, they tend to hear more frequently about the small number of potentially harmful microbes that are a natural part of those communities, in that newspapers and television programs will tend to focus on cases of outbreaks or disease, whereas there are very many other microbes in the environment that are doing other things that can be quite positive, and others that are not having very much impact at all. So context really is very important. So these are just some examples of different niches within food systems where microbes play a key part. I should have pointed out that when I talk about a microbiome, that's essentially the community of the various different microbes and the different components that are found in that environment. And so you will see individual examples of those microbiomes here and where they can be found. And then subsequently, Paula will tell you about the interconnectedness between these. So you will see that a number of these are marked to highlight the potential for microbiomes contributing to disease, such as relating to consumption of shellfish with particular pathogens present therein, or plant disease, or animals, or diseases in humans. But there are very many other examples where microbes on or in food systems having a very great beneficial impact on those individual niches. And indeed, those niches couldn't survive without the contribution, the important contribution of the microbes present therein. So I was originally trained as a classical microbiologist. I did my PhD working with different agars and broths, growing microorganisms, looking at them under a microscope in order to study them. And there was the potential for that to continue, but had it done so, we would have been quite challenged in terms of trying to fully appreciate the full complexity of microbial communities and what they're capable of doing. And the reason for that is that very many microorganisms do not grow, or it's quite difficult to grow them in the laboratory. And so if we rely exclusively on growth-based approaches, we will only see the tip of the iceberg. There is no one agar, there is no one broth that will grow every microorganism. It does not exist. And so in the absence of such an agar or such an approach to facilitate growing the microorganisms and studying them, we need an alternative to harness those difficult to access microbes. Now, these approaches were originally developed for environments such as the human gut, where we know that very many of the microbes present therein require certain unusual situations like the absence of oxygen or the presence of bile or other components in order for them to grow. In the absence of knowing in each case how to grow those microorganisms, the alternative that has been developed has been the use of DNA sequencing-based strategies. So what do I mean by those? So if we can extract the DNA from an environment, all of the DNA from all of the microbes present in an unbiased manner, we no longer need to grow the microorganisms, but rather instead we can analyze the DNA sequence data to determine what microbes are present and what they're capable of doing. So when we isolate all of the DNA from all of the microbes from a particular environment, we refer to that as metagenomic DNA, and we analyze that DNA to get a better understanding as to what microbes are present and what they're capable of doing. And so in that regard, I'm going to provide an example or some examples of what we in the lab and subsequently on our computers do when we want to study these various different microbiomes. So one approach involves amplicon sequencing. Amplicon sequencing is kind of the equivalent of generating a barcode for representing the microbes that are present in a microbial community. So amplicon sequencing can come in different flavors. The example I'm going to refer to here relates to 16S or RNA sequencing. And this is a type of amplicon sequencing that's used specifically for looking at the bacterial component of a microbiome. You can also use other approaches like ITS sequencing to look at the fungal component. So using the 16S gene as an example, there are a number of features associated with that gene which explain why we use it. So first of all, that gene is present in all bacteria. So we can then, as a consequence, study all bacteria using this approach. The gene also has features whereby certain regions within the gene are conserved and other regions between those conserved areas are variable. The conserved regions allow the use of a PCR-based approach where we need the knowledge of that sequence data for what's referred to as a PCR primer to bind. And that amplifies the region between those two conserved regions. And the subsequently generated variable data or variable regions can then be compared with a database. So that database would have existing information regarding to 16S sequences from very many other bacteria, and we can identify what microbes are present, what bacteria are present, typically to the genus level. And that's an important consideration because the genus level will tell us whether a Listeria monocyte, whether a Listeria is present or a Bacillus is present, but it won't always necessarily distinguish between a Listeria monocytogenes and a Listeria innocua, which is quite important because one can kill you and the other is harmless. Increasingly, as the field has moved on, there has been a greater focus on an approach that's referred to as shotgun metagenomic sequencing, which no longer focuses specifically on just one gene type, but rather tries to sequence as many different genes from an environment as possible. So again, once you have a good approach to efficiently and in an unbiased way extract DNA from that community, You can then subsequently sequence it in an untargeted fashion, hence the term shotgun, and from the genes that are sequenced, get different types of information. One, again, relates to the taxonomy or the names of the microorganisms present. And importantly, in this instance, it allows you to get down to species and even strain level identification. So you really can determine whether you have a problematic or a desirable microbe present within that community. Indeed, you're not just focusing on bacteria in this instance. Because of its untargeted manner, you can also get information not just relating to bacteria, but fungi and viruses, including bacteriophage and so on and so forth. You can also get information regarding the various different genetic pathways that are encoded within these different microorganisms. So how many genes associated with toxin production or antibiotic resistance or some desirable feature. And then finally, if you have enough data and the particular microorganism of interest is present in sufficient abundance, you can actually generate enough data to create what's referred to as a metagenome assembled genome, or a MAG, which in effect gives you the equivalent of a genome for a microorganism that in some cases has never been isolated in a laboratory. So you know a lot about what that microorganism is capable of doing in the environment, even without ever having cultured it, which is very powerful. So to give some context and to explain as to why the field has really evolved so considerably over the last number of years, is to maybe look at the technology that generates the sequence data. So again, presuming you can efficiently extract the DNA from the community, or you can extract RNA and convert that to cDNA for analysis, then subsequently the challenge becomes how much analysis, how many bits of DNA that you can sequence. So in sequencing parlance or sequencing terminology, we refer to reads. So these are regions of DNA that are sequenced. And there are a number of different types of sequencing technology. There are those that generate small bits of DNA sequence data, which I guess if you could maybe think of these as small pieces of a very large jigsaw. And for the sequencers that generate small bits of sequence data, the focus over time has been to generate more and more pieces of those jigsaws to give us a complete picture of what it is that we're looking at. So over the last 15 years or so, the technology has moved such that the original cutting edge instruments, such as the old Roche 454 sequencers, which used to generate 1 million bits of jigsaw, have now moved on. And so there are now technologies that will generate up to 20 billion sequencers, or bits of sequence data. There are other bits of jigsaw that will generate much longer pieces of jigsaw that are valuable for other reasons that I won't get into now, but it's good to have an appreciation of the various different technologies. And so as a consequence of the development of these new microbiome-based approaches, we have the opportunity to move away from some of the culture-based approaches that in some instances can be slow or inaccurate. They may only detect one or a small number of target microbes at a time. And in some cases, and quite often, you need to know what you're looking at in advance. In contrast, using microbiome-based approaches, you can study all microbes regardless of whether you expect them to be present or not in a community. There are options to use portable DNA sequencers as time has gone on, and they can be very accurate, but it is important to have good databases to facilitate an accurate description. And so using these approaches, it's possible to understand the contribution of microbes and microbiomes in food systems as they pertain to safety, spoilage, flavors, and also health attributes. And these are all things that you'll be hearing more about as we proceed today. It could have been argued in the past that for certain environments, such as specific foods like cheese, we already know a lot about the microbes that are present therein because there was a presumption that we could sequence all of the microbes that were present, or we could grow all of the microbes that were present. However, one example that really, I guess, made me appreciate the full potential, I guess, of microbiome analysis relates to a study that we carried out maybe over 10 years ago now, where we were trying to determine the basis for a spoilage defect that we see in a certain type of cheese. And we tried all sorts of different microbiology-based approaches without success. And it was only as a last resort when we used DNA sequencing-based approaches to look at the microbiome that we identified a certain type of microorganism from the genus Thermus that was present in the cheese that had this discoloration defect that was not present in the control. And through that sequencing-based approach, we were able to subsequently grow that microorganism, a microorganism that had not previously ever been associated with a cheese environment, but is more usually associated with hot thermal springs like those you would find in Yellowstone Park and elsewhere. So as it turned out, armed with this information, we were then subsequently able to go back to the cheese manufacturing facility and found that indeed this microorganism is present specifically in the hot water systems of that plant. Okay, so hopefully I've given you a taster for what's to come. I'm going to pass you on to Rui now so that we can proceed.
Thank you, Paul, for the great introduction and providing a foundation for today's discussion. And our next speaker is Dr. Paula Fernández Gómez of the Universidad de León, Spain, where she is a Beatriz Galindo Junior Researcher, and she's recognized for her innovative contributions to food safety and functional food development. Paula has a background in novel strategies for biofilm control in the food industry, employing non-thermal atmospheric plasma technologies. Recently, her research focus has shifted towards the development of functional fermented plant-based foods, utilizing metagenomic and metabolomic characterization techniques. After obtaining her PhD in veterinary and food sciences from the Universidad de León in Spain, she joined Teagasc Food Research Center through the prestigious Margarita Salas postdoctoral fellowship, followed by an Inspire co-found Marie Curie Fellowship. Paula, thank you very much for being here with us today. The floor is yours.
Thank you very much, Rui, for the introduction. Well, yes, after this introduction, I'm going to be guiding you through some examples of microbiome interconnections and some interventions that can be applied on different environments along the food system. So I will be starting with primary production environments. There you go. And I'm going to be talking from the soil and from aquatic environments, rivers, oceans. In the case of soil microbiomes, we know that there are shared species between microbiomes of soil, roots, and also the aerial part of plants. And we also know that they are influenced by many different factors, and these effects can be both short-term and long-term. So we can harness all these factors in order to develop novel strategies to apply on these environments. Many of these studies actually come from just observing nature, especially in extreme conditions. We can observe how these microbiomes are different and identify certain microorganisms that can bring us some benefits. That's why I'm going to talk about a couple of examples of how microbiome-based interventions can be applied to crops. For example, there's certain isolates, certain bacteria that have the potential to be added to soils in order to improve the phosphorus solubilization or the nitrogen fixation ability of the crops and their productivity. Also, there are some isolates that can inhibit the growth of pathogens, and in this way, we could be protecting certain crops from disease. This could be applied not only to crops, but also in land for pasture, for livestock animals. For example, there are studies focusing on intercropping strategies that have a microbiome base underneath. And the theory is that this could help nitrogen fixation efficiency and in general improve the yield of pasture. Also, there are strategies using certain isolates that can enhance the resistance of pasture to drought, which is quite important, especially with all the issues related with global warming. Moving on now to aquatic environments. Here, the microbiome study has two very big roles. The first one would be to first understand how agricultural practices can impact these environments, and also to develop novel strategies to repair ecosystems that have already been damaged. Some of these issues that I'm talking about, for example, is the runoff of fertilizers to rivers that can arrive to oceans, or, for example, the same runoff of antimicrobials or antimicrobial resistant bacteria. One environment that is especially susceptible to anthropogenic stressors are corals and sponges. And actually, there are some studies that are trying to identify some beneficial microorganisms that can be used to recover this ecosystem. If we continue moving along the food system, so yeah, we move on now to the post-harvest processing and distribution environments. Here I want to first talk about fruits, vegetables, and cereal production. In this case, microbiomes are, sorry. Microbiomes are really important because there is a very close connection between the microbiome present in the plant when it is in the field and when these type of products, fruits, vegetables, arrive to the consumer, especially if these two places are close together. It's just so that there is even a term that is edible plant microbiome. Also, these microbiomes are very sensitive to the storage conditions during, for example, distribution and the packaging. And this is important because there is an estimation that up to 50% of the fruits and vegetables are lost during this stage, during the post-harvest stage. And again, there are several opportunities for microbiome-based intervention at this stage. The first one is the mapping of food processing industries. This is possible thanks of novel technologies like stream resolved metagenomics. What this allows us is to identify specific strains, not only species, but strains inside a food processing plant. So we can identify the point of entry and kind of follow the route that they follow to different places, even to closer to the final product. And this is especially interesting because it can help industries predict sources of bacterial contamination or identify certain points that need more attention inside the food processing plant. And another application, another intervention that I wanted to talk about is the use of certain bacterial isolates in order to postpone or decrease the post-harvest decay in fruits. A good example is strawberries. This type of protected cultures could be applied in order to improve the shelf life of a product. Moving on now to terrestrial farm animal production. This is a sector that is expected to grow a lot in the following years, especially if it's going to meet the predicted demand. And here there are many different examples of microbiome interconnection. A very studied one is the microbiome in the gut of production animals, but also there is a close connection between the microbiome of the feed. It could be pasture, it could be silage or other type of feed, and the gut microbiome of animals. Also, livestock grazing is going to have an impact on the soils, rivers around these production farms. And of course, we need to remember that at the end, these microbiomes are going to have some microorganisms that are going to end up in food processing plants. They will enter through the raw ingredients. In this case, there are also many different examples of microbiome-based interventions that are being developed. Just to highlight some examples, for example, there are some interventions that can be some isolates that can be added to feed in order to modify the gut microbiome and in this way reduce methane production. Moving quickly through aquaculture, again, different examples of how microbiome is important here and how there are many different examples of interventions that are being developed, like monitoring fish skin microbiome, predict population collapse, again, the use of probiotics supplements in diet to improve the health of the animals. And finally, talking about retail and household environments. Here is also important to understand the role of these microbiomes in, for example, spoilage and food loss. And I wanted to touch very quickly on the role of bacteriophages. We are learning more and more about bacteriophages and their role in shaping microbial communities. This is, again, thanks to the development of, for example, high-throughput DNA sequencing technologies. For example, this allows us new applications that are in a very early stage. For example, we are starting to understand the role of phages in fermentation failure. And just to finish, to talk very briefly about the impact of these interconnections, microbiome interconnections on human health. And just with these omic technologies, we are understanding more the relation between our diet, the gut microbiota composition, and there is more and more evidence of these transmissions of microorganisms from the food that we eat and the microorganisms that we find in our gut. At the same time, we are starting to realize that we are distancing ourselves from the food microbiota. This is because of all the treatments that need to be applied to food in order to be safe and to have large-scale industrial production. But there are also some microbiome-based interventions that are being explored, as I said before, protective cultures to improve the shelf life of products, but also we are understanding more and more about probiotics, prebiotics, and bioactive compounds, and also about how they are present in fermented foods naturally.
Okay, so our final presentation is from Dr. Tanja Kostic, a senior researcher, engineer, and thematic coordinator at the AIT Austrian Institute of Technology, as well as a managing director of the MicrobiomeSupport Association. Tanja's research centers on the microbiome of diverse ecosystems, particularly food production environments, where she develops and applies innovative methodologies and technologies to preserve and utilize microbiome data. With extensive experience in fostering collaborations between research and industry, Tanja is recognized for her efforts to standardize practices and policies in microbiome research and for advancing microbiome science by promoting both innovation in food and biotechnology sectors. She also played key roles in numerous European projects such as Master, Domino, or Food Safer. Tanja, thanks again for being with us today. Over to you.
Thank you, Rui, and thanks to Paul and Paula for setting so wonderfully the food systems microbiome stage. What I will try to do in this very last part of the webinar is to somehow show how do we come from understanding towards the impact that we will all like to see, and also what still hurdles and gaps we have in front of us. So to start, I mean, Paul already said how sequencing technologies contributed more and more to understanding the microbiomes. And there are different approaches of how we can address the microbiomes and different data that we can collect. So if you look at the one hand on the metagenomics data, and there was a lot told about the metabarcoding and the shotgun sequencing, it tells us a lot about who is there, the taxonomy, also about the functional potential of the microbiome. But what it doesn't tell us is really what the microbiome is doing, about the activities, who is doing what, how they're interconnecting. So if we dive deeper in the omics technologies using like metatranscriptomics or metaproteomics, we will start to see really specific activities of the individual microbiome members under investigated conditions. And this is important to understand because our experimental setup or how we sample or what we investigate will have an influence of the answers that we are getting. Microbiomes are extremely dynamic systems. They react to the environment, they interact with their holobionts. So situation will change and we're always looking at a snapshot in time in a way. Paul also told us a lot about challenging in culturomics and getting down to this isolate, to the bacterial or fungal or archaeal isolate. But in the end of the day, we will need them for developing many applications, especially I come from Austria, a GMO-free country. If you cannot use gene technologies to use some of these functions, if you want to do really a sort of natural environmental applications, then we need to sort of come back to the isolate and have the isolates in our hands. Nice thing is that the more data we have, the more we understand from the metagenomic data, it's easier to tailor the isolation conditions so we can get more and more isolates that we don't have yet. I will also show you a little bit how this is developing. So this is just a snapshot from August this year in the availability of the data. And this is not a complete snapshot. This is just what's available in the Magnify database. This is the biggest European database of the different sequencings. There is more out there, but I think that it gives a good snapshot. So what you see as an amplicon, and don't forget, amplicon is just telling us who is there, is sort of 10 times more than what we have in the metagenomes. They're giving us this complete functional potential. So there is already a big discrepancy there. It sometimes also costs, it's still a question of cost, of accessibility of the technologies, of a complexity of a microbiome in a certain ecosystem where sometimes we cannot go deep. And then if you look at the right type, at the biome type, you will see that we have a lot of data sets from humans and also from digestive tracts. But then if you look at some specific environments like plants or even food production, that there are less and less data sets. So it's not equally distributed. And if you want to go even deeper into discussion, if you look into the human microbiome, we will see that there's like geographic representation is not there. So we also here have huge gaps, even though the data sets seem more comprehensive than with the other biomes. Also to highlight the other gap, and we talked a little bit about this agar paradox and agar problem. This is from a publication that was published in 2021, and you see on the left the bacterial and on the right side the sort of fungal tree of life. And I would just like you to zoom on this sort of numbers on the right side of the taxa, where you see how many actually cultivated isolates we have. And you will see in many, many groups that we don't have these numbers, indicated that we don't have a single isolate on our hands, meaning from the metagenomics data, we have these MAGs, we know the organisms, we have classified the organisms, we know what they can do, but we don't have them and we cannot experiment with them, and we cannot really go into development of applications with them. So these are the big gaps that we still have to address, and this is something that the community is working on. And there was perhaps a phase where we were focusing almost solely on sequencing. We are understanding now that everything needs to go hand in hand if we really want to go and achieve an impact. Paul touched also a little bit on, once you have the sequencing data, you have to do a lot of analysis on the computer, a lot of bioinformatics analysis. And then on the one hand, there are also multiple dimensions to consider. So on the one hand, we have this microbiota, all microorganisms, everything that we know about them, but also then this theater of activity. So what's really happening? What do we see through other omics technologies? How do we integrate this different data set? So from the microbiome, we have developed these different ecological concepts. We talk about the core microbiome, we talk about keystone taxa, we talk about indicator taxa for certain conditions. This is a knowledge that we can get out of that. But then in order to make full use of this data that we generated, think back on one of my first slides showcasing the magnifying data set, we need data to be fair. So they need to be findable, accessible, readable, and interoperable, but they also need to be standardized. So if everyone's using a little bit different method, if we don't have all of the metadata, if we don't even know how the data was generated and we know how volatile the microbiomes are, then it becomes more and more difficult to reuse this data set. And this is now a big effort of the community, is really going into this sphere. How can we make data really fair? How can we sort of use a certain baseline of metadata standards that we can make use of the data in the future? And that's not like one data set per study that's being generated. And also in the food area, there was a huge effort where sort of a curated food metagenomics database was generated some years ago, which really helped us understand food system microbiomes in much better way than we did before. And then, we are living in the era of artificial intelligence, and artificial intelligence and different machine learning methods are really helping us to integrate and understand this multi-layered data, because we have the microbiome data, we have the omics data, sometimes we'll even have an environmental or phenotypic data. So we really need a high computational power, but also new methods to really to find the interpretation, to find this interconnectedness, and to be able to make sense out of our data. We also need to think about application developments, because just because we understand microbiomes doesn't mean that we can immediately develop a microbiome-based solution or application. So we usually start at the laboratory scale. So we know there is a potential in a certain microorganism. We can test them in a quite simple experiments, but then we need to transfer to the models, test in the greenhouses, test in vitro systems, test in animals, laboratory animals, and then we have to go to the into the real world. And as we go down this funnel, the complexity of the conditions that are sort of this application in development is facing is getting more and more complex. It's more and more difficult to control the conditions, and it's always important on the one hand to really work on the development, but also to apply the methods that we sort of discussed at the beginning to monitor what is happening in the system. So we need to address, as I said, environmental conditions. We need to look more deeper always in the interaction between the host or environment and the microbiome and the application that we are trying to develop. And we also need to see, and Paula showed it so nicely, the transfer across the system. So if I develop, for example, a biofertilizer or a biocontrol agent for the agriculture, for growing of the crops, how does this application influence the quality of the food that's produced? And when I eat the food and it comes into my gut, how does it interact with my gut microbiome? So those are important questions, and we are just starting to tackle them, as I said, because system is so complex, and we are just getting the tools to address these. There are many potential applications. So we discussed about improvement of food systems, about more sustainable production when you're thinking about, for example, fertilizers, about improving the resilience of the agricultural systems that are facing climate change, increased productivity. There is also potential to use the microbiome-based application to restore the ecosystems that are deteriorating. We know, for example, that we are losing, the soil health is deteriorating, and without healthy soils, there won't be really a lot of healthy food in the future. We can also use it, as Paula said, to track the movements of the specific strains in the environments, also to track, for example, outbreaks. And in this review, we have brought together many, many applications developed by numerous groups. It's really a community effort. It's also what our review draws on. And for the interested people here, just dive in. There's so many great examples of what has already been done, first success stories, and something that we can really build upon in the years to come. We also need to think about how this application develops. So, and we have some years ago discussed this. So what's really a microbiome-based application and how we can categorize it. So there is clearly this easy, sort of this straightforward, untargeted effect. If you change agricultural management, if you swap to, let's say, Mediterranean diet, you will have an impact of the microbiome in soil, in your gut. You will see the changes, but we call these effects untargeted because those are natural responses of the microbiomes, and we cannot really tailor them in a way. So we don't know how successful they are. We know there are changes, but we don't know how they are. We are moving now more into the direction of a semi-targeted effect. So it should be like, if we have a sort of a universal microbial consortia that can help support plant growth promotion, or, we have now many of these probiotics out there that most of us will get after antibiotics or together with antibiotics to buffer the effect of antibiotics and help our gut. Again, they're semi-targeted. We know the basics, why we are using this. So knowledge base of the condition when they're applied and what is applied is higher, but we are still in a semi-targeted effect. And where we are hopefully moving into the future is what we call precision application. And that would be then really to say we don't have this universal plant growth promoting consortium, but we have a plant growth promoting consortium for soybean that's grown under, for example, in soils that are poor in nitrogen or phosphate. Or we have a probiotic mix that's applied after we have tested the gut microbiome and know there's a specific dysbiosis there. So this is, as we said, we start with a low level of understanding and low probability of success. And we are moving now towards this higher level of understanding and accordingly higher probability of these solutions having a success and showing an impact that we want to show. Moving forward, I also want to emphasize it's not only about research that still needs to be done. Yes, the research needs to continue. Yes, there are many things we still don't know. There are many microorganisms that we still don't have. So there is much more that has to be also invested there. But the system is so complex and we have to also consider other elements. For example, the microbiome is never mentioned when we're talking about the protection of biodiversity. And you still have to imagine and keep in mind that every time we lose a higher organisms like a rare orchid or a nice butterfly or whatever, we use its complete microbiome. So thousands and thousands of organisms associated with this one. We also know that even though, for example, research collaboration within the EU works really well, and it works partially internationally, but we exchange information, but we don't have funding to run, or we have limited funding to run international collaborative projects. So this is something that needs to be improved because as microbiomes are interconnected, we are, and we need to all work together. I already discussed a little bit about how to improve the use of big data and the possibilities of AI, but also the needs for fair data. So this is something that we need to further work on. And then we need to continue developing application technologies. So if you think applications of the microbiomes in the field or applications of the microbiome in the food processing, these environmental conditions that the microbes have to face, we need to somehow protect them. We need to give them the best chance to establish themselves, to do what they're supposed to do, what we are using them for. So this is still one of the issues that we have to tackle. But we have also more soft issues. The regulatory environment. It doesn't help that we as a scientific community and together with all of the great research done within the industry have a solution ready if the regulatory environment is not tailored to them, if the products cannot be registered, if we don't know how to register them, how to apply them. We need to work all together on managing expectations. Sometimes I hear so wild story about what not applications of the microbiomes we have, and some expectations are really, we are done, the research is done, we have all solutions in our hand, we can save the world. No, we are part of the solution. We still have many questions, and we need somehow to also manage these expectations, not only within the community, but towards our funders, towards the end users, towards farmers. We really need this clear communication and really set the scene properly, that the people are not disappointed and that we have a future. And the last element that's really important for that is to increase the microbiome literacy in society. So I really love what Frontiers Hub did also with this little program for the young readers, etc., because it's so important to share this message realistically, to share the knowledge about the microbiomes and to really reach out also outside our community. We also need to stop thinking and working in silos. Microbiomes are connected. We have to be and stay connected. So if we want to really achieve this impact, then we need to work much more together, exchange the information, harmonize the method. And this is the only way how we can really make sure that the microbiomes can be used and can make their impact on human and environmental health. We know the potential is there. We just need to pave the way for exploiting this potential. And for that, we need everyone on board. We need a scientific community. We need food producers. We need regulators. We need educators. We need the consumers. And I also hope that this review and the hub and all which was done now would also help a little bit to promote this amazing system of the food systems microbiomes, and will help us to go one step further or several steps further and into the hopefully more bright future and more impactful future.
Thank you, Tanja, and for taking us through the journey from understanding to impact. And we really hope that this lead article and all of the hub content. It's one of the initial steps, or one of the middle steps, because there was a lot of research already done in the past, like to achieve that objective of bringing everybody out of their silos and into a more sustained conversation, I guess, and towards the future. So I hope all of you attending are enjoying today's webinar. So far today, we have learned from 3 experts and learned quite a lot about agri-food system microbiomes, their interconnections, and potential applications and impacts. It is now time for us to move to the panel discussion, and I'm delighted to welcome our 2 guests. Our first guest panelist is Karel Callens, Senior Advisor in the Food System and Food Safety Division of the UN Food and Agricultural Organization, FAO. Karel is an international leader in microbiome science and innovation, and throughout his career, he has championed collaboration between scientists, innovators, policymakers, business leaders, youth-led startups, and civil society, fostering inclusive and cross-sectoral dialogue. Something that we really, like Tanja said, we really need in this conversation also. At FAO, Karel has played a pivotal role in strengthening the science policy interface and has been instrumental in advancing the organization's work on the applications of microbiome science and innovation for sustainable agri-food system transformation. In his current role, Karel supports the FAO's mandate as custodian of Sustainability Development Goal 2, helping ensure that the world's poorest and most vulnerable countries benefit equitably from advances in microbiome research and technology. Our second guest panelist today is Julie Barrett O'Brien, CEO of the OpenBiome Foundation, a global organization dedicated to advancing microbiome science and patient outcomes. Julie builds impactful institutions at the intersection of science, policy, and public health. She has been a vocal advocate for investing in health systems strengthening and played a pivotal role in the successful resolution on universal health coverage at the United Nations. A distinguished lecturer at Yale's Jackson Institute for Global Affairs and a guest lecturer at Boston University School of Public Health, Julie has also served as an advisor to prominent organizations such as the Clinton Global Initiative and the World Bank. Welcome to you both, and thank you for joining us today. Okay, we will start with a couple of questions for Karel and Julie, and then we will open up to questions from the audience. So please continue to submit your questions to the Q&A tab, and thank you for that. So Karel, I would like to start with a question for you, if that's okay. So the lead article maps the intricate and interconnected nature of microbiomes throughout the entire agri-food system, from soil to human digestion. Given your strong emphasis on integrating microbiome science into the One Health framework, ensuring that low and middle income countries are at the center of innovation and policy efforts, how does this comprehensive understanding of the agri-food system microbiome interconnections help inform the FAO strategy for achieving sustainable and equitable food security and health outcomes, particularly in these low and middle income countries where microbial diversity is rich, but the One Health risks are disproportionately high?
Thank you, Rui, and maybe let me start with congratulating the authors of the paper. I think it's a very fascinating paper. It's unique that it brings all the different aspects of agri-food systems together in one paper, and really emphasizes the centrality that microbiomes play across the different aspects of the food system. That said, let me maybe start with saying what FAO is. So the Food and Agriculture Organization of the United Nations, it's an intergovernmental organization, and the majority of our member countries are actually low and medium income countries. So that's why we think it's very important to think about those countries from a food systems and a microbiome perspective. We are not a research organization, but we are very much interested in research because we very often operate at the interface between science, policy, and practice in terms of agri-food systems. And so it's obvious then that we're interested in microbiome science, still a very relatively new science, but to see what the policy implications are of this, also at an international level. So we have a strong focus on SDG 2, the Sustainable Development Goal 2, which focuses on food security, nutrition, and sustainable agriculture. And so I think we heard from the presentation of the three speakers how microbiomes are central to SDG 2. And so for that reason, we are paying a lot of attention to this. But it's not only central to SDG 2, it's also central to One Health. And One Health is a really interesting concept. It's still relatively new, I think, in the policy circles. And it has been spearheaded by FAO together with the World Health Organization, UNEP, the United Nations Environmental Program, and the World Animal Health Organization. And the idea is really that the health of people, plants, animals, soils, water bodies, the environment in general, they're all interconnected and interdependent. And so we can't think about trading off the health of one, maybe human health versus against environmental health. Both really need to be valued. And the interesting part, and we heard it from the presentation, is that actually microbiomes are really an integrator. So they integrate health across these different spheres. And this is why we are starting to talk about actually microbiome stewardship. So microbiomes are not just microorganisms by their own, but it's an ecosystem. And it's part of the bigger, so it's a micro ecosystem, part of a bigger ecosystem. And so in the same way that we think about valuing the large ecosystem, the macro ecosystem, we also have to start to value the microbiome ecosystem in itself. Maybe that sounds obvious to the people in this audience, but I can tell you that among policymakers, this is a very alien concept. A lot of people actually don't really understand what microbiomes are. They don't really understand what a pathogen is within a microbiome and how you can actually, through the management of the microbiome, control these pathogens. So a lot of the thinking is still very much focused on individual microbes. And most of the attention, and when you think about One Health, is on the pathogen. And we forget all the other organisms around that and the valuable role that they play. So this is why we talk about microbiome stewardship, to say it's a really fundamental piece of biodiversity. And I think Tanja mentioned it. This is not recognized as yet. I mean, scientists talk about it, but policymakers don't. It's not being monitored, not nationally, not internationally. And it's really important that we start to think about that. Now, why are low and medium income countries important? Not just for us in FAO, because they're the majority of our member countries, but also because it's where the large majority of the world population is located. And we've talked about food security, we've talked about ill health, not just malnutrition in the form of undernutrition, but all the common non-communicable diseases, obesity, etc. And we know that these are linked to microbiomes. But also these countries are really where most of the agriculture production is taking place. A lot of the food that we consume in high-income countries actually originates from there. So they are a major source of commodities that are getting exported. And then also they are a large reservoir of biodiversity. I mean, a lot of these countries still have areas that are not touched yet or maybe minimally. And so there is also this biodiversity aspect and the microbiome diversity that is there. So it's a large untapped reservoir that leads into innovation. So there's bioprospecting that actually taps into that. And so we need to think about that as well, and what the benefits are that also these countries get out of that. And so when we think about agriculture in these countries, we need to think about two types of agriculture that coexist. So what I talked about is export of commodities. So you have large scale industrial agriculture that by and large actually applies green revolution technologies. And we all know what the impacts are of these technologies in terms of environmental damage, etc., but also damage to the microbiomes of soils, water bodies, etc., to pollution, etc. And then we have a very large sector of smallholder farmers. Actually, the large majority of farmers in these countries are smallholders. And so the issue with smallholder farmers is that it's very low tech, low input, and there's enormous productivity gap there. And this is where we think that, so from the smallholder perspective, that actually microbiome applications, and I'm happy Tanja talked about the targeted, semi-target, and precision ag. Well, these are farmers that definitely would benefit from targeted and semi-targeted technologies, microbiome technologies, to bridge that gap. While actually the industrial agriculture could probably benefit from more precision approaches.
Great.
Maybe just to come to a final point here is that so much of our work actually is around at this point in time is the normative work. So we're looking at integrating microbiomes into global frameworks. And one of them is actually the One Health Joint Plan of Action. So we've recently released a paper on that, looking at the 6 action tracks of the One Health Joint Plan of Action, and with concrete examples on how microbiomes can be integrated in there. But I'll leave it at that. Thank you very much.
Great, thank you. And I think that was a really important point because it connected with what Tanja was saying before about the lack of including this microbiome in the most of the documents, unofficial documents. So I think it's a good step. So thank you very much for that. And now on to you, Julie. Thanks again for joining. And in your policy outlook, you advocate for a holistic microbiome-informed approach to agri-food systems and childhood nutrition, stressing that food is medicine throughout his interaction with the gut microbiome. Considering the Leeds article detailed insights into how microbial ecosystems interact across the broader agri-food system and their profound influence on human health, how does this enhanced scientific understanding of microbiome interconnections strengthen the OpenBiome Foundation strategy to develop and implement equitable microbiome-based solutions for childhood undernutrition?
Thank you very much, Rui. And I am honored to be here with all of you. I continue to learn every day from listening to everybody. And so I thank you all for sharing your points. And I'll be short because I think a lot of folks have said, made some excellent points that I would have made. And I know we want to get to the questions, but just to provide a little bit of context about the OpenBiome Foundation, we got our start at the Massachusetts Institute of Technology as this unique spin-out, turning healthy people's stool into a life-saving treatment called a fecal microbiota transplant for patients with a horrible gut infection called C. difficile. That was our mission, to bridge that gap till products were approved, and that happened. So we were the key thing here, and I think this is related to the microbiome discussion today, is although this fecal transplant worked, they don't know exactly why. They just know it worked to 80 to 90% efficacy. And that's what's so fascinating about the microbiome, is that we can come up with these treatments potentially. And I think this has given some false hope to potential future microbiome-directed opportunities because nobody's been able to duplicate the success rate of what this FMT did for C. diff. But that intrigued us and that fascinated us. And so when our mission was accomplished, we evolved into a foundation to take our resources and our know-how and to make some investments in key areas. We're a nonprofit, so small grants focused on pilot concepts that will get future funding, bigger funding, that is addressing some of these unmet needs. And child health to us is a really important piece of this equation, and trying to better understand the gut microbiome of kids and its impact on obesity, which I believe the UN just released a report that said there are now more obese children in the world than there are undernourished, which is, I think, coming from the United States, I can attest to that. Here. So it's a real issue that we think needs more investment and more attention to it. The second piece, the equity piece, is really important, and I'm glad Karel spoke before I did, because the FAO is doing some amazing work here. And the idea that, at least from the human gut microbiome, and I can't speak to the agricultural as well as everybody else on the field here, but 86% of the data that is publicly available is from guts in the United States or Western Europe. Now, we know that is, I think it represents 14% of the US global, or the world population, which is completely biased, and we consider that wrong. So we are making some strategic grants in more of the developing countries to try to increase the amount of data that comes from there. and so that we can get it into a more open source data, so we can get it into the to equalize the opportunity for all people, because everybody deserves to benefit from science. So from an equity perspective, we see that as really important. One of the early partners we had with this wonderful group called the Global Microbiome Conservancy out of Kiel University with Doctors Mathilde Poyer and Mathieu Grossin. I don't know if anybody from their team is on here today, but kudos to them for leading that effort. And I do think it's an opportunity to even leverage these global collaborations. They're focused on identifying the normal healthy gut microbiome worldwide. But what's to say you couldn't overlay some agricultural influences into that? I mean, they've got the platform. Those are types of things where we can really take advantage of existing programs. So thank you. And I'll leave it so y'all can get to the good questions here, into the deep science, which, by the way, I'm not the one to answer those questions.
Great, thank you, Julie, for helping me with the time, and thank you to Karel also for your initial thoughts. And we are now opening the floor for some questions from our live audience. Just as a reminder, we have a bit more than 20 minutes until the end of the webinar, so if we can keep the answers as short as possible, even with the lots of content in them, I think that's a good challenge for all of us. So think of it as a quick fire round of questions. And yeah, we'll start right away. So our first question, we had a lot of interest in the theme about harnessing microbiome innovations for sustainable food and nutrition. So the first question for Paul and for any other that speakers that wants to reply afterwards comes from Barry Bruce. He says that we are working to create a sustainable local food system prototype based on regenerative agriculture principles. What are the practical and affordable ways to measure the health of our soil? Are there any specific indicators or simple methods you could recommend for small-scale community-based projects?
So I'm not a soil expert. I think Tanya's the soil expert on the call, so I might just direct immediately to Tanya rather.
Yeah, go ahead, Tanya.
Yeah, thanks. Can you hear me? Yeah, okay, because
Yes.
my bill. I mean, the big question of healthy soil. That's the I think it's just still as big as the question of healthy gut. There was an interesting policy paper by the EU that published some healthy soil parameters. Unfortunately, microbiome was not mentioned one within this document. So there are obviously a community efforts in trying to revise that and say, okay, we have to look at the microbial diversity. I think it's really now difficult for, these small communities, because we are still learning what a healthy soil is. So we don't have like, a quick dipstick kind of essay. And this is where I want to sort of debunk the myths, because I think there's always, we always say the more diverse the community, the better, but for that you need a certain depth of analysis. So I think we are learning all together. What we are trying to do in our research institute, and I would like to see that more in the future, is really more of these farmers project, more of these lighthouse projects within the EU, where we can really, as a scientific community, go out into the real fields, test, amend the data, and then provide this knowledge and information back to the community. I think that's the way in front of us, and not a quick test that's available right now for the farmers or initiatives.
I should have said we at Teagasc and the Vista Milk Initiative, we have some citizen science projects ongoing, where the approach has been to give the citizen scientists some cotton briefs, some cotton underwear, and they're putting those in the soil and then digging them up a number of weeks later to see how successfully the microbes can break down the cotton. And that's a very crude indicator of the success or how healthy the soil is.
Great, thank you, Paul, for that additional information. And citizen science is very important. I think that like together with the open science, it will help a lot, like to advance what we are advocating for. Another question that we had about this theme from Sadia Latif, it's Also, to any who might want to answer this, but particularly directed to you, Tanja, also, how can the microbiome in crop fields be engineered to improve agricultural outcomes? What are the potential long-term impacts, and is there any risk for soil pollution associated with the microbiome engineering?
So I think, first of all, to start from the back, there is no risk-free technology. And I think we need to learn. So what was just recently discovered, that bacillus was often used as a biocontrol, and then some strains that are perhaps not so safe for food came into the food. So we really need to be careful when accessing risk and when interacting in the system. Engineering the soil microbiome is difficult due to its complexity, and the best results are perhaps to try to engineer more towards plant. So through different, because at the end of the day, you know, it's a plant that's in the center probably. So there are different products or also application technologies, how the microorganisms around the plant in the rhizosphere or within the plant can be engineered for the better plant health. It's always important to monitor the things like establishment, spread, and all of so everything, if there is sort of inoculation or if we are trying to introduce the new microorganism, then a proper and full risk assessment should be done and not disregarded.
Great, thank you. The next question is for the authors, but so I'll start with Paula. Also to give you the chance to answer. Your lead article extensively maps these interconnected microbiomes, describing how they can be traced across soil and marine environments to human gastrointestinal niches. Could you elaborate on one specific example where a disruption in one part of the agri-food system microbiome, like the soil or the aquatic environments, as you mentioned in your presentation, has a direct and significant but often overlooked consequence on human health?
Okay, very direct. Okay, I can think about maybe some examples, but just to highlight that it's more about the link between the successive microbiomes, the different environments. So the first one affects the second one, and the second one affects the third one. From soil, one example that comes to mind is how an altered microbiome can maybe affect the ability of crops to absorb certain minerals. Then at the end, we will be including in our diets these plants, these fruits that maybe are lacking some of these minerals. Maybe there are some other examples Tanja, Paul can think about, other examples of direct relations.
Yeah, one that I often use as an example relates to the production of fermented foods through spontaneous fermentation. So spontaneous fermentation is where the microbes that are naturally found in that environment contribute to the fermentation process. So something like sauerkraut. So the microbes on the surface of the cabbage leaves will be determined by the environment in which they're grown, but also subsequently after harvest where they're stored. And then the subsequent fermentation process, you add salt and that enriches for certain microbes. But if you don't have the right microbes there to begin with, or you introduce a pathogen or a spoilage microbe somewhere along the way, you might get a food that is not, or a sauerkraut that is not produced properly. And that can result in spoilage issues, or potentially in a worst case scenario, even the introduction of a food pathogen along the way. So you have multiple different environments, from the soil to the surface of the plant, to the storage area, to the fermented food, to the human gut.
Great, thank you, Paul. As we move to the questions, one of the points that a few of the attendees raised was actually in relation to one of the points that the authors had discussed with us as one of the important points for this discussion today, about the need for networks to better link scientists in the field. And one of the questions comes from Afalake Olambiwinu. I'm really sorry if I pronounced your name wrong. And his question is, as a food microbiologist that has been working on fermented foods, I have been trying to work on the microbiome of fermented foods that are indigenous to us in Africa. With no modern facilities to carry out the research work as adequately as it ought to be, how can researchers in developing countries partner with the global north to achieve this? And this was directed to Karel, and then anybody else that wants to add any thoughts are welcome to.
Yeah, that's a good question. I mean, we know that the infrastructure, not just the infrastructure, even the knowledge actually about microbiomes, also in education systems, etc. I mean, in many of these countries, this is not yet covered. So one of the things we are looking at is really cooperation, South-South cooperation or triangular cooperation, so between low and medium income countries and high income countries. And the establishment of networks, basically. And this is actually a core function of FAO as well, where we convene players across all these countries to discuss about issues, but also to do capacity development, etc. So I must say in the microbiome field, this is still very, very early days. But discussions are taking place. So the example I gave you on this framework paper that we're working on is an example where member countries from across the whole world will start to discuss about microbiomes. And hopefully then that will lead to policy change, including investment in the research and partnerships. I think also, and maybe Tanja, you can talk about that, but in the context of Horizon Europe, the initiatives often involve also countries from those regions. Yeah.
Yes, thank you, Karel. So there are some, I would say, also limited opportunities for within the Horizon Europe, especially for collaboration within Europe and Africa. But raising this international network is so important, and also having the funding to support this international network is so important. But I think we need to also keep in mind, and we have been working in scope of different initiatives also on the ABS, so access and benefit sharing plans. So there is a huge diversity. We know that, for example, let's go back to these traditional fermented foods. They're so different than what we have in Europe. They're so rich. They're such a source of potentially, microorganisms with the interesting function. So if we want to, go there, reach and exploit these things, then it's also our responsibility to give something back in terms of training, in terms of collaboration, in terms of data sharing. So I think that community there, scientific community, is really now trying to acknowledge that and also trying to set up a proper framework condition. We do need just simply more, I would say, formal ways, more funding ways where we can include the scientists from the global South also, like South American, so to strengthen the collaborations and to really be able to, provide them, because we don't always need a new sequencing facility. There's so many around. You just need to, open the access towards what's already there. And there are several microbiome initiatives now. There is a MicrobiomeSupport Association, the World Microbiome Partnership, and we are all trying really to work on that, to interconnect the scientists and then raise this voice together also towards the policymakers and said, these are the things that are missing. These are the things that we need to address in the next policies, in the next regulations, in the next fundings.
Great, thank you. Our next question I'm gonna direct to Julie, just because it comes from I also have kids, so childhood is very important for me at the moment. So Julie, the OpenBiome Foundation champions the equitable innovation and addresses this childhood and nutrition. What are the specific mechanisms or funding models that are most promising for ensuring that the microbiome directed therapeutics and interventions currently that are often prohibitively costly, they are very costly for the in high income countries, become affordable and accessible in low and mid income countries, effectively addressing the needs for the vulnerable populations?
So of course you asked a really hard question.
Yeah.
Like how to make food really cheap? I think this has been a big challenge with RUTF and the ready-to-use therapeutic foods, which are life-saving, but they don't necessarily repair, children's tummies that have been, their gut microbiome that may be disrupted from a number of different issues and diseases. And I think that that's one of the things we're trying to work on right now. We've actually, so speaking of global collaborations, we've been speaking with Jeff Gordon and at Washington University and Tamid Ahmed at ICDDB. I always get that mixed up at the in Bangladesh, and about this idea of Jeff's done some fascinating research and Tamid on microbiome directed food. And particularly they're interested in weaning. But the idea is that you would try to find locally sourced, locally developed foods. And this is where the cross-sector research really is helpful, because the health folks are very focused on the gut microbiome and its impact and functionality. They don't understand soil. They don't understand how can we improve the crops, how can we improve the nutrients within those crops that might be able to positively feed the gut microbiome of these kids. But also knowing that everybody's gut microbiome is different around the world. You can't just have one size fits all for everyone. You have to localize as best as you can. Can we do that precision health for every single person? I don't think that's realistic. I think Dr. Kostic, you had mentioned let's be realistic in the beginning in your presentation. I think that's important. So, but truth of it is locally led, locally developed, and culturally acceptable and adaptable is the way you're going to get to better costs. Because I think if we're just looking at developing some product elsewhere and importing it in, that's not going to work very well. So you have to start with cost in mind at the very beginning of the process with the right people at the table.
Great, thank you,
Could
Julie.
I come here for a second? I think this is a really important policy question in the sense that a lot of these countries, low and medium income countries, actually the food base, the food basket is narrowing and it's westernizing. And actually there is very little promotion investment in indigenous crops. And if you start to look at these legumes and fruits and vegetables, etc., so the fiber-rich foods, but also traditionally fermented foods, I mean, with that, at the low cost, actually, you could produce a microbiome-friendly diet. And I think this is something that's completely off the radar. So we're talking about importing these ready-to-use therapeutic foods, etc., while actually these people sit on the resources with which they could do that. So to me, this is a big policy question. Thank you.
Thanks, Karel. Okay, so we're almost at the end of the webinar, so we have time for a couple of quick questions. The next one is directed to Paul from Shweta Ran. Since not all microbes can be grown in agar in the lab, the database of all microbes cannot be fully comprehensive. Could you please explain more about how the sequences are used in database formation under these circumstances?
Yeah, I'd point to the curated food metagenomic database that we were involved with in generating a number of years ago. It was published last year, but just to very briefly explain what was done in that instance. So if you sequence a food microbiome, or indeed any microbiome, you will get all of the DNA from all of the microbes that are present if you extract in a non-biased way, as I mentioned earlier. That DNA will come from the microbes that you can culture, but also a number of microbes that are difficult to culture or cannot be cultured also. And if the microbes that cannot be cultured are present at sufficient abundance, so they're at high enough levels in that sample, and therefore you can get enough sequence data relating to it, you can actually create the equivalent of a genome, a circular bacterial genome or a fungal genome from that data to give you the equivalent of a genome that you would get from an isolate on an agar plate. So quite often, we know a lot about what certain microbes can do, even without ever having grown them, because we know they have pathways associated with using a particular sugar or producing a particular metabolite. And also it's very clear where it fits in the phylogenetic tree of life.
Great, thank you, Paul. Okay, so unfortunately we run out of time for today's discussion. I see that we still have a lot of questions that need to be answered, but hopefully this is the beginning of a beautiful continuation of the work, and we can discuss this issue and its improvements and how we move forward with the discussion in future opportunities. But before we leave, I want to ask for a final takeaway message from all of the panel. So if you have a final comment or action point, something that you feel that it's really important for all of the people that are joining us today to take, either from a relevant scientific community, relevant agencies, policymakers. So Paula, if I may, I'll start with you, and then I'll ask the rest of the panel for their thoughts.
Yeah, I would say just to remember to treat the food system as a whole, to remember that all the microbiomes are interconnected, they vary over time, and just how these omic technologies can allow us to understand microbiomes better, and therefore to develop novel strategies for a better, more sustainable food system.
Great, thank you. Karel?
I would say microbiome literacy. I think it's the biggest impediment to uptake of this science and innovation. So if we can all contribute to that in a scientific manner, because there's a lot of also not so scientific information out there, but I think that would be enormously helpful.
Great, thank you. Tanja?
Yeah, I think it's really to manage expectations. So let's communicate what we know, but let's also communicate what we still don't know and where research. So let's not be afraid of admitting that we are not at the end of the journey.
Great message. Julie?
Sorry. Yeah, I want to go back to something Tanja said at the beginning, which is the standardized data. And if we have that across sectors, then we can share more, share better, and create better policies from that. But if we're all operating in silos, we're not going to get very far.
Great, thank you. And finally, Paul.
Maybe to extend Julie's point, the importance of teamwork or collaboration from right around the world, and that's not just from different scientists in different fields, sometimes literally in different fields, but also the regulators, policymakers, industry. And so it's going to take an approach from multiple different angles, but people working away in isolation, not caring or not wondering about what others are up to, are only going to be able to make so much progress.
Great, thank you for your final thoughts. And okay, we're almost at the end of the session. I would like to thank all of today's speakers and expert contributors, Professor Paul Cotter, Professor Tanja Kostic, Dr. Paula Fernández Gómez, Karel Callens, and Julie Barrett O'Brien. Thank you very much. To all of you around the world, thank you for joining us today. We hope you enjoyed the session. Before we close today's session, make sure that you visit the Frontiers in Science website to read the article we discussed today and its related content. You can continue the conversation on social media using the hashtag Frontiers Forum. Be sure to subscribe to Frontiers in Science updates. The link will be posted on the chat, and visit the Frontiers Forum website for upcoming events. So for now, from me, our guests, and the entire team at Frontiers, we say goodbye. Thank you once again and for joining us, and until next time. Bye-bye.